With your PPE on, start by wiping down your entire bench with 10% diluted bleach. This is an initial cleaning to disinfect the workspace, so make sure to wipe down the pipettes, tip boxes, tube rack, benchtop trash bin, and all other surfaces. Repeat this process with 70% diluted ethanol to get rid of any residual bleach, as bleach will degrade plastics over time. Carefully shake out lysis tubes onto a clean Kim wipe and label the tops and sides with the sample ID. Next, open both caps and place upside down on a Kim wipe. Use a 1000 microliter pipette to pipette 800 microliters of DNA-RNA shield into one tube, and repeat this process again for your second tube. Make sure your samples are thawed and labeled. Take out a plastic knife, making sure not to grab the tip. Mash up your sample from outside the plastic bag until homogeneous. Push up a small specimen of fecal matter to the top of the bag. Depending on moisture level and debris contained in the sample, take care to section off an amount that is free of visible debris. Use a plastic knife to separate out a pea-sized amount of fecal matter. You may need to scrape the knife against the top of the tube to remove it. Transfer this amount into your prepared lysis tube. Discard your plastic knife and change gloves as you switch between samples. Again, make sure to wipe down your bench area with 70% ethanol in between samples, in case any of the sample fell on the bench. Make sure to change gloves before switching to another task. Carefully place your samples in the vortex, ensuring they are balanced on opposite sides. Turn on the vortex. It will shake for 30 seconds, breaking open cell walls within the sample. Turn the vortex off and carefully remove the samples. Samples should be visibly liquefied. Discard all generated waste into its appropriate bins. Decontaminate your bench space following protocol from the beginning, wiping down all surfaces and lab equipment with 10% bleach. Make sure to allow for 15 minutes of contact time with the bleach before wiping everything down with ethanol. As a reminder, instructions may vary on your specific lab equipment. Ensure proper training before getting started. This video is brought to you by the Cartsenal Lab 4A video production team at Brown University.